All right, let's go to the answers on your homework here. Number one, Nick, what do the letters DNA stand for?
Something nucleic acid.
Deoxyribonucleic acid. Okay, does anybody remember what that deoxyribose thing refers to? Specifically.
The type of sugar that's in
Type of sugar that's in the nucleic acid, right? So we're going to later study RNA, which is ribonucleic acid. We'll look at the differences, but the basic difference between the two is just a single oxygen that changes the sugar. We'll see how that works, but it makes a big difference to how they act within inside your cells. Okay. Riley, what two scientists are generally given the credit for figuring out the structure of DNA? Watson and Crick. Franklin was instrumental, but she didn't really figure out a structure. So they're the ones, I mean, you know, when I talked about it yesterday, I don't mean to take any credit away from them. They're the ones that figured it out. But they couldn't have done it without Franklin's analysis. So Franklin was important in it, but they're the ones that actually put two and two together and actually figured it out. Anybody remember the name of the other person involved there? Wilkins. Bruce Wilkins. All right, number 3 says DNA is a polymer, which means it's made up of many repeating single units, which can be called monomers. What are the monomers called here? This is a type of organic molecule. Wait, Maya. You're close. The nitrogenous base is going to be part of this thing. What are the nitrogenous bases part of? The nucleic acid or the nucleotide. Right? So we tend to call them nucleotides. So the nucleotide is the thing that's made up of, as you see up here, the sugar, the phosphate group, and the nitrogenous base. So the nitrogenous base is just one part of the whole nucleotide. Okay? And you can think of it as being the most important part because that's what creates the bridges in between the two DNA molecules. The whole nucleotide is the backbone of the DNA molecule is made up of 2 components. Katie, what would those be? The sugar and the phosphate. Way good. Okay, so remember, it's sugar, phosphate, that's what runs the rails of our twisty ladder. Okay, there's 4 different variations, 4 different bases. What are the names? No, give me one. Guanine. Okay, Nick, another one? Adenine. Isaac? Thymine. Okay, and Rachel? Cytosine. You need to know those names, but pretty much from now on, we can't just refer to these as G, A, T, and C. So I do want you to know the names, but we abbreviate those all the time, so there's nothing wrong with that. Doing that in your worksheets and stuff. Okay, now I kind of skipped over Chargraph. Chargraph did an experiment where he showed that adenine and thymine, there was always the same amount of those two, and there was also the same amount of cytosine as guanine always. So based on this information, we can tell that A pairs with T and G pairs with C. Okay. Part of your homework tonight is going to be looking at a replication of DNA, but you have to match up those different bases. So this is called complementary base pairs or base pair rules. One strand of DNA is complementary, and it should be opposite to the other strand. they oppose each other. I mean, they're bonded, but they oppose each other. If they were matching, it would be A with A and T with T. Okay, and then the bases are paired by what type of bonds along the center? Weak hydrogen bonds. You can't just say hydrogen bonds. But it is important that they're weak, so that the DNA molecule can separate from the. We'll be talking about that today. Okay, any questions there? All right, once Watson and Crick had figured out the structure of the DNA, then scientists started going, well, okay, if it's a double helix, we know it's going to have to get copied. Because for a cell to make a copy of itself, to go through mitosis and have the chromosomes separate, it first has to copy the DNA. All right, here's a little Who remembers what part of the cell cycle the DNA gets copied in?
S2.
S phase. There isn't an S2.
It's S phase.
It is an S phase. So remember, the cell cycle is G1, which is normal cell stuff. S, which is where the DNA is copied. G2, where it kind of builds itself up, and then mitosis. So remember, the DNA replication, where you get new chromosomes, has nothing to do with mitosis. All mitosis is about is separating the chromosomes. So in the S phase, we make new DNA. And there was three kind of theories on how that might happen, three hypotheses. One is called the semi-conservative model. In the semi-conservative model, they said, well, what if each strand of the DNA kind of gets pulled apart, and then you get a new strand that gets built on, each of those individual strands. So what happens there is the two new cells are going to end up with kind of half of the DNA that was originally in the parent and half that was completely built new.
Just wanted to let you guys know at 3:10 you're going to be dismissed to go to the Commons to wish Marie farewell. Mrs. Stevie and her sisters are going to sing a song, and then we're going to sing We Wish You a Merry Christmas. Wish you farewell. So,
Gotcha. Good job, Ray. Yeah, right.
All right, let's focus, please. Okay, now, when we look at this, we need to realize this is not talking about, like, in meiosis where the things get split up. These two forms of DNA, or these two chromosomes right here, are still going to be identical to each other. But the idea is to copy copy itself. We're going to split half of the DNA strand up and kind of build a new copy using that in each of the cells. So that's the semi-conservative. The conservative model says, well, maybe the DNA just gets completely copied and one cell ends up with the original molecule and another cell ends up with a completely new molecule that's been built from scratch. Okay? And then there's what's called the dispersive model. And some people said, well, there's really can't see a reason why necessarily all that stuff would stay together. Maybe it just gets all chopped up and then put back together. And so each new cell is going to get just a conglomeration of part of the chromosome from the parent cell and part of this brand new put together. Okay? So those were the theories on, and I shouldn't call them theories, they're a hypothesis. It's a hypothesis on how it could happen. Scientific theory is a little bit more solid than that. So to study this, what they did is they took the parental DNA and they basically added a bunch of amino acids that were going to be radioactive. So they knew that any new DNA that was built would have these radioactive substances in it. And then they analyzed the new cells. Okay? If the conservative model was true, one of the new cells should be non-radioactive, and the other new cell should be radioactive because it's built out of all the new DNA. In the dispersive model, we should see kind of partial bits of it. In a semi-conservative model, we should see each of the cells having the amount of DNA in them as well, or the radioactive DNA. So the first thing that they did is they let the cells do that, and they realized that each of the new cells have some radioactive DNA. So that right there rules out this. The conservative model can't be true because both of the new cells have radioactive DNA inside. But they realized if they let the cells go through another division, and they looked at them, if the dispersive model was true, then all 4 new cells should have radioactive atoms inside. If the semi-conservative model was true, then here half of the cells would have non-radioactive DNA, half of them would have radioactive. And what they found was the semi-conservative model was the one that stood up to the scientific analysis. So we realized what's happening inside your cells when you make new DNA is they're actually using half of the DNA as a template for one cell, half of the DNA as a template for the other cell. Right? So we know that these two are not the way that cells copy their DNA. Follow the semi-conservative model. So let's take a look at that a little bit more in depth. Now we talked about yesterday how the DNA strands are linked together in the middle by those weak hydrogen bonds. To be able to separate them and make new DNA pieces, the first thing that has to happen is we have to take those 2 strands apart. And that's where a special enzyme called helicase comes in. Okay? So the first thing that's going to happen, and we'll write that down here, we'll kind of write some steps. So this is how DNA replication happens. The first step is that DNA helicase separates the 2 strands. Okay, and helicase is just the name of a specific enzyme. Remember, an enzyme is just a special protein that can do chemical reactions. But that enzyme starts taking apart, breaking those hydrogen bonds, and taking apart the strands and separating them. Okay? Now when it does that, we get kind of this area in here where the helicase is working, where the 2 things are still stuck together and they're slowly coming apart, and we call that area a replication fork. So the replication fork forms because the DNA helicase is separating the strand of DNA right in there.
So that's part of step 2.
No, that's part of step 1. That's just the name of the area where step 1 is happening. So step 1 is DNA helicase separates the 2 strands. Okay? Once those strands are separated, at that point, then we can start building the new strand by putting new nucleotides in. But that takes a different type of enzyme, and that enzyme is called DNA polymerase. Step 2 is that DNA polymerase starts adding new nucleotides. So it starts building a new strand. It does that by adding nucleotides. Sorry, my C and L got a little close to each other there. That should be nucleotides, not nucleotides. So, kind of like just snapping Legos in a line, this polymerase grabs a nucleotide and adds it to the chain. Moves over, grabs another one, adds it to the chain. But it has to add them correctly. Because the whole point here is that A goes with T and G goes with C. So it has to grab the right one and add it on so that the chain can be built. Otherwise, they're not going to bond together correctly. But because A and G and all those things only go with each other. What we get is identical strands. Because here we had an A and a T that were together. When they get separated, this A is going to bind to a new T. This T is going to bind to a new A, and you end up with two strands that are identical to each other because of the way that they're being built. So you're going to have a DNA polymerase working on the top and the bottom strands at the same time. This one's going to be putting in whatever opposite one that this one's putting on in that same area. The way they're built. Okay, does that make sense? Did I go up too fast? Put that again. Okay. Now, there is some other stuff that happens there, like the DNA polymerase can't really start until something else has been put on. We're not going to worry about some of those little details. We're just going to hit the broad strokes. So the helicase separates the two strands. The polymerase starts adding new nucleotides. Now, step 3 is that we need proofreading. Okay? The DNA polymerase is pretty good about putting the right nucleotide in there, but every once in a while it does screw up. And so there's these enzymes that come along and actually check its work and make sure that, yes, this is an A and there's a T bonded to it. Because every once in a while something messes up and you might have an A with a C bonded to it that's not bonded right because they don't match up right. So that has to be removed and replaced and fixed. So proofreading and fixing, and that is going to happen due to a bunch of different enzymes. So we're not going to worry about the names of all those enzymes that work on the proofreading. But that's an important thing that happens. If that didn't happen, every time you copied your chromosomes, there would be some screw up in there. And screw-ups we call mutations, and mutations tend to be bad. Now, we're going to find out that when we study stuff later, after Christmas, there are some mutations that still get through. There's some mistakes that are not caught in the proofreading step. But a vast majority of the mistakes are found and fixed. We'll look at what happens, what kind of mistakes can happen, and how it affects the cell. But I think we've already talked about what does a mutation usually do to the cell?
Kill it.
Kills it. The cell can't survive because of mutation. So in most cases, that's what happens. But we'll look at some of those things after Christmas. So those are kind of the 3 main steps. The helicase separates to 2 strands. The polymerase starts putting new nucleotides down, and as it moves along, there's a proofreading mechanism that comes along to make sure that the nucleotides that are there are the correct ones. Okay. Now, those are kind of the broad steps. Of course, there's a lot of complicated things that happen there. Well, there's a couple of things that we need to talk about that have to do with how the strands are actually built inside the DNA. Okay? Just want to make sure everybody's caught up there before we move on. Any questions? All right. Now, if you remember yesterday, we talked about the fact that DNA is built anti-parallel, right? It has directionality. One strand goes one way, the opposite strand goes in the opposite directions. Kind of think about your arms going like this, right? Remember, we call that anti-parallel, the fact that they're parallel to each other, but they run in opposite directions. The thing is, when we look at how DNA is replicated, the DNA polymerase can only go in the 3 to 5 prime direction. Okay, and when we say that, we're talking about the original strand. So here's one original strand. The dark blue are the original strands in each case. In this case, this is 5 prime. This is 3 prime on this side. That would mean that this here is 5, and this here is 3. On this strand, the DNA polymerase doesn't have any trouble. It hooks on, it starts building the complementary strand that starts building the DNA on the other side, and it just does it on and on and on and on, one after another. No problem. We call that side the leading strand. So the leading strand is the side of the DNA where the polymerase can just build it continuously. Okay? On the other side, we have a bit of a problem though. The DNA helicase is separating the two parts of the DNA molecule up here, but the polymerase can only move in this direction on this side because it always has to go from 3' to 5'. So what that means is the DNA polymerase here, when it attaches, it's going to build along until it hits a point where it's already built some. And then because it's opening up back here, there's more real estate back here where it can do some work, it kind of leapfrogs backwards and builds some more along until it hits where it started there. And then it leapfrogs backwards again. We call this the lagging strand. Okay? Now because of that, because of this leapfrogging by the DNA polymerase, it can only work in one direction, that means you get these little chunks of DNA that are built kind of one at a time, and they're not really connected to each other. We call these little things Okazaki fragments.
I think the guy who found them was Asian.
Japanese. Yes. So the Okazaki fragments are the little chunks of DNA that get built kind of one little bit at a time as that DNA polymerase leapfrogs backwards. But the problem is those things are not connected to each other. And to be able to form a DNA molecule, they need to be connected to each other. So here we have another enzyme called DNA ligase. The ligase comes in and it connects the Okazaki fragments. Ace on the end of a molecule means it's an enzyme, and so it just depends on what it does. Helicase is called that because it unwinds the helix. Polymerase is called that because it makes a polymer. It attaches monomers to a polymer. The ligase is called that because to ligate something needs to attach.
Was that for the... Is that a U?
That's an A, sorry.
No, after the G of fragments.
On fragments? That's an M.
Oh.
So the DNA ligase just attaches all the fragments, and now you have a nice continuous run on the other side. That only happens on the lagging strand. Right, Isaac?
Yeah. I'm awake.
You are.
Dude, I was so confused.
I am awake.
No way.
I have a headache.
I thought I agree. We all agree. The thing is, he'll try to comprehend further.
It's no big deal. I understand it's the afternoon. Just ate dinner.
I'm tired, but I was awake. I heard everything.
Yeah, you should just give us a 7 because we don't understand any of it.
Anything that ends in ace is an enzyme.
Listen. Let's talk about what you're going to need to know. You're going to need to know what each of the enzymes do. Okay? So on a test, I might ask, what does DNA helicase do during replication?
Separates the units.
Yeah, it separates the two DNA strands. I might ask, what does DNA polymerase do?
I won't say that again.
It makes polymers.
Okay, hold on.
You
Sorry.
guys stop answering the questions.
Yeah, I was the one sleeping.
Yeah.
I understood. I'm not saying okay. What did you say, Tyler?
What enzyme separated?
The DNA helicase. So the helicase right here is what separates the two fragments.
Just do it. Separate.
The two strands.
Okay.
Okay. So that's one. There's 3 enzymes here. So that's one. The second enzyme is DNA polymerase. It's the one that puts the new strand together. It builds the strand. One nucleotide at a time. And then the other enzyme that you need to know is DNA ligase. And the ligase attaches the Okazaki fragments. Now again, these fragments are just built because the polymerase can only work in one direction. It can't go this way. It's not built to go this way. It has to go this way. But because of the way this is separating, you only get, stuff on this side. And so it's going to jump back, build some, until it hits this part, and then there'll be more of it opened up where it can jump back and get some more. But because of that, all these things are just tiny little chunks of DNA. They have to be bound together to be able to be a DNA molecule. And that's what the ligase does. The ligase comes in and attaches them, kind of welds them together so that they're one long fragment. Again, that only happens on the lagging strand, because the leading strand it cannot be in that direction. Okay, does that make more sense now?
Yep. Okay. I'm ready for the test.
Yes, Nick. Now, what's interesting is the DNA helicase, the one that separates the chromosomes, does not just start on one end. I mean, you might think, oh, okay, here's the strand of DNA. The helicase is just going to start on this end and work all the way to the other end. But it actually kind of starts in the middle. And you're going to get replication that goes in both directions.
Okay.
Yes, I'm teaching. Oh, yes.
Merry Christmas.
Sorry, thanks. Okay.
Twice.
Like they're confused that I might be teaching during our class period.
Yeah.
So, the DNA is going to get replicated in both directions, okay? Here's what's really happening. It starts in one place, but you're going to have one helicase that's going this way, opening it up. There's another helicase that's going this way, opening it up. So where you have a leading strand is kind of flip-flop depending on which direction the helicase is working. So here you'll have a leading strand, here's a lagging strand as it gets built kind of backwards. So in that case, you have kind of this bubble that keeps spreading out and out and out. And in fact, on a single DNA molecule, you're going to have lots of places. If you just had one single replication site that moved across the entire chromosome, it would take forever for that chromosome to get replicated. So what we actually have is hundreds of different sites all being worked on by a whole bunch of crews. It's kind of like building a bridge, right? If you had one crew that came in and built a little bit of the bridge, and then the next part of the bridge, and then the next part of the bridge, until it was all the way over, it'd take forever. What they do is they bring in a bunch of people. They start building from both sides and meet in the middle. It takes a lot less time. And that's what's happening here, although we have lots of different places. So we have these replication bubbles that spread out as the helicase opens it up and build new strands with the polymerase until they meet each other, and then you get, once that happens all the way across the molecule, then you have two new chromosomes. And then you can go on to G2. And mitosis, you get new cells. Okay. So those are the basic things that you need to know about DNA replication. Again, the whole point is just to copy the chromosomes so each new cell, after you go through mitosis, can have those. Or in meiosis as well. Either way, you have to copy the chromosomes first. But those are the kind of things that you need to know. So the different enzymes, what they do, kind of the overall process of how they go together. Okay? Now, I have a little video that we're going to watch. It's going to show this animated, so that might help you kind of see how it works. I will say a couple things about this video before we watch it. There are a few more details in the video that we need to worry about. So we're going to talk about some stuff that we didn't talk about. Don't worry about that. Number two, the way this video was made, I believe they made it by, you ever been to one of those websites where you can just type stuff in and then it'll talk it back to you?
I'll try Google Translate.
Well, yeah, but there's other websites like that. So some of the pronunciations are going to be slightly off, but it should still show you what's going on.
DNA replication. At the end of this module, you will be able to describe the process of replication. DNA replication can be defined as a process in which an entire double strand is copied to produce an identical set of DNA helix. The process of replication starts at a specific point on the DNA. This point is called the origin, or origin of the replication site. The first step in replication is the binding of the enzyme helicase to the origin. The function of helicase enzyme is to unwind and separate the strands of DNA. The strands of DNA are complementary, and the moment they are separated they tend to bind back. This is prevented by proteins called single-strand binding protein, which binds to single strands of DNA and prevent their rebinding. Now, a second enzyme called the DNA polymerase binds to the separated strands and begins the process of replication. But DNA polymerase can add DNA nucleotides only to a pre-existing chain of nucleotides. Therefore, replication really begins when an enzyme called RNA primase assembles a small strand of RNA called RNA primer at the origin. The RNA primer is a short stretch of RNA nucleotides which are complementary to a small section of the initial sequence of DNA strand. DNA polymerase is now able to add DNA nucleotides to this primer and initiate the process of building a complementary DNA strand. Once a complementary DNA strand is built, the RNA primer is removed by enzyme action and appropriate DNA nucleotides are added. The two strands of DNA are oriented in opposite directions. But the polymerase enzyme can carry out replication only in one direction. Therefore, a different mechanism is used to replicate the opposite strand of DNA in the second strand moving in the opposite direction. DNA is replicated discontinuously to produce a series of discontinuous DNA fragments. These fragments are called Okazaki fragments. As the Okazaki fragments are built, the RNA primers are replaced with DNA nucleotides enzymatically. The individual Okazaki fragments are then bonded together to form a continuous complementary strand by an enzyme called ligase. The continuously formed strand is called the leading strand, and the strand synthesized in small pieces is called the lagging strand. The process of DNA replication is completed when DNA ligase joins all the short stretches of DNA into a continuous strand.
Alright, you done it.
That almost confused me more.
Twice.
3 times. Because he said DNA twice, and then there was DNA. All right. There's your question of the day. I know we're not writing it down, but you can at least think about that. For your homework, you're going to do 8.1, although I apparently screwed up, and the packet that I gave you does not have the second part of 8.1. It only has the first part.
Oh, darn.
So that kind of backfired on my whole hand out all of the worksheets at once plan. So let me come around and I'll give you the second part of 8.1. I'll give you 12 parts for homework for Monday, January 6th.
Sugar and nitrogen.
I don't want to do it.
I could carry you some.
I got plenty of time right now. Let's get that started. I will say there's lots of good videos on YouTube about DNA replication. There's ones by Crash Course and Bozeman Science and stuff. It wouldn't be a horrible idea in January, Sunday night, before you come back, take 10 minutes and watch one of those. 10 minutes is not a lot of time. It'll remind you of some stuff, and you won't be near as lost when you come back to school. So, think about that. Also, don't forget, when we come back, you only have like 2 weeks to get your DNA model done. So it might be a good idea to get started on that, at least get your materials gathered that you're going to use to put it together. If you need another rubric, go on the website, find it.